just said, I'm the MRI Scanner Operator at 7T, so I spend most of my time either at the scanner computer or scratching my head over the resultant data afterwards. Oopsie daisy, there we go. Whoa, I accidentally gave you guys quite the spoiler there.
Yeah,
okay.
That's a quick talk. Thank you for your talk today.
That's all for today. Yes, so the project, one of the main projects that I'm involved with is kind of stretching the definition of animal here, but it's an ex-vivo human spinal cord injury project. And that is run through a number of organizations. It's I think spearheaded by Brian Kwan, who's a neurosurgeon here in Vancouver. And it's called the International Spinal Cord Injury Biobank, which basically has the goal of filling the gap, the human-shaped gap in spinal cord injury research. So it's a global repository with data available to researchers across the world, and we also have participants enrolled from all across North America. So because human tissue is so valuable, we wanted to take full advantage of that gift. So as you can see, there's quite a number of sample types and methods that are used here. Obviously, my area of involvement is with the scans. So we scan the donated tissue on our 7T Bruker BioSpec scanner, and we're using a birdcage style volume coil, which has an inner radius of 35 millimeters. It also has a 4.5 centimeter range along the Z-axis before you start to see signal drop off. So to accommodate that, the cord is cut into 4.5 centimeter pieces after it's excised, and then we're given the cord in those pieces. So this is a little snapshot of how it's going so far. There are almost 300 participants, and so far 14 cords have been donated. You'll see that those 14 cords are represented in this diagram at the bottom, which is very bright and colorful. And we just use this to keep track of how our workflow is going. So anything that is red or orange means that I have work to do as far as either scanning or mold design goes. And you'll see as well that each cord is divided into those 4.5 centimeter blocks. So the process for this project after extraction starts with 2 weeks of fixation in formalin, and then the cutting into those segments for us. Those 4.5 centimeter segments are brought to our facility where we run a battery of scans, kind of roughly outlined here, and I'll go into more detail in just a minute. And then using those scans, I design our molds for histology, which is shown in a diagram here. This is kind of what the current design generally looks like. And the goal for that is to have those 3 millimeter segments aligned with the protocols that we're using. So the slice positioning and orientation for our protocols. The pieces, once cut, are embedded in paraffin, and then they can be sectioned and stained for histology. So we run, as I said, a kind of a battery of scans. We start with some anatomical images. The flash sequence is the one that is used as a reference for designing the mold, although the high resolution rare image is also useful for that, just because it shows features slightly differently. So sometimes in the flash, there's a little bit of ambiguity around sort of where the tissue ends, especially if we're looking at an injury epicenter that's very damaged. So the rare is useful for that. We also run about 2 hours worth of diffusion scans, and then do some IHMT and QT2 as well, which Piotr was just talking about. So this is sort of a little bit of an overview of what those scans end up looking like. This slice that I'm showing you is an injury epicenter from one of the more recent donors, and you can kind of see that the structure has been changed by the injury, and that, you know, obviously has implications for various measures of diffusivity as well as myelination. So we run a 6-shell diffusion protocol. You can see that we have b-values ranging from 500 to 7,000. And then we also take 2 A0 scans with a b-value of 0. One of those is reverse phase encoded so that we can do some distortion correction. And we subject that data to quite a few diffusion models. And again, Piotr touched on some of those. The ones that will come up later in my talk are DTI and DKI, which were both used in correlating some of our MRI data with histology. So as Piotr said, we use IHMT, which is just an MT technique that looks specifically at myelin. So that dual saturation in the protocol allows us to sort of break down the coupling between the Zeeman and dipolar order pools. And so then we're singling out the dipolar order to get an image that's more specific to myelin. And I've just restated this equation here at the bottom, which Piotr also shared. And here we have an image of IHMT ratio maps that were calculated using our 2 types of IHMT protocols that we run. One is T1D filtered, and the other one is cosine modulated. And again, this is the same injury slice as we were looking at a little bit earlier. We also run a myelin water fraction. Well, we run a QT2 sequence, which gives rise to our myelin water fraction map. This sequence is about 1 hour and 8 minutes long. And I've borrowed this diagram here from Alex McKay and Corey Laula, who are both heavily involved here and in this project, just to kind of provide a bit of a visual of how the myelin water fraction is found just by looking at the areas under the T2 curve at different time points. Yeah. So actually, one of Corey Laula's PhD students who just defended, Sarah Morris, she did some work comparing MRI and histology, which is very much in line with some of what Piotr was talking about. So she was looking at myelin loss in cords that were from this biobank. So the myelin content with MRI was assessed using our myelin water fraction, IHMT ratio, as well as DTI and DKI. Specifically, she found correlations with fractional anisotropy and radial diffusivity. For histology, she was looking a Luxel fast blue stain, and then also comparing that with some of these molecules and proteins that are associated with an immune response. So Sarah found that the myelin water fraction and IHMT ratio were strongly correlated with the Luxel fast blue staining, which is good because that validates these imaging measures or methods rather as biomarkers for myelin, which just means that we can use them hopefully in a clinical setting, although again, some of the concerns about IHMT have been raised in terms of timing. But yeah, this will be helpful just for clinical decision-making and that kind of thing. Sarah also found correlations between fractional anisotropy and radial diffusivity, correlating with the presence of, again, those proteins and such that are associated with an immune response. So was looking into those as biomarkers of inflammation. And then here are also a couple of other papers that have been related with the biobank. I won't get into those. What I will get into is sort of this act of correlating MRI data with the histology, which, as Piotr pointed out, can be quite a challenge. So I think it was probably my supervisor, Andrew, and my predecessor, also called Andrew, who came up with the idea for these molds, which is, as I mentioned, to align the pieces that are used for histology with MRI slices. So as you can see, we have sort of a series of slots along here, and those are placed at 3 millimeter intervals, which is just to accommodate both. We have a diffusion scan, which is 1 millimeter sliceness, and then our HMT and QT2 scans, which are 1.5, so that allows both of those. And in short, what I do is I take the anatomical images we have, segment them. 3D Slicer has a wonderful option to create a 3D mold using this, or a 3D model. So I create that 3D model and then bring that in. Oopsie, sorry about that. Bring that into Fusion 360, which, oh dear, which is just a 3D design project, and then use the subtraction of that model to create this mold. So that's kind of what this process looks like. And yeah, that's everything from me.